Studies chromosome structure, number, and behavior. These are all things related to the genetics. This type of information used in karyotyping, diagnosis of chromosomal abnormalities, and in the modern concept of the taxonomy, we are using cytotaxonomy. Cytotaxonomy is nothing but based on the cytological information. We can use the information in classifying the plants at order level or at a family level, at a genomic level or species level, how the chromosomes are there, shape of the chromosome, behavior of the chromosomes at the time of meiosis and mitosis, and the size of the chromosome and shape of the chromosome, karyotype, and we are all, this type of information comes under cytogenetics and clinical and medical genetics. It is applied genetics completely to diagnose and prevent and treat the genetic disorders. It includes the genetic counseling, parental testing, and gene therapy. You know very well, genetical disorders are there so many. How these disorders are prevent, and how we replace the correct gene in the place of mutant gene. When any dispute between the parental and the infant at hospitals, we can use the parental DNA testing, genetic counseling also. So that is nothing but is called Menarika Sammanchina Vivahala Nappudu. We can give the genetic counseling also. That is comes under clinical or medical genetics. So nowadays we are using genomics word. It is a buzzword in the modern biotechnology. Study of the entire genome, all genes of an organism, includes structural genomics, functional genomics, and comparative genomics. So basis for personalized medicine and genomic editing also. For example, based on the genomic sequence, we can design a proper drug. The drug is only meant for the particular person. So in this aspect also, we can go for the research work that is comes under genomics. It is completely entire genome of an organism. We know all the gene structure and we know all the gene sequence also, and all the gene size also we can determine by using advanced technology. One more is epigenetics, is that study of heritable changes in gene expression that do not involve changes to the DNA sequence. What are the heritable changes are in gene expression only, not in the changes in the DNA sequence. So it is influenced by environment or lifestyle and development. So this play key role in the cancer, aging, and stem cell research. And one more branch, that is pharmacogenetics or pharmacogenomics. So it is study how genetic variation affect the drug response. So once you visit the hospital, doctor prescribed a particular antibiotic. Sometimes the antibiotic not work properly. We can go for alternate another drug. Alternative antibiotics are there. We can use alternative antibiotics, how they are respond to the particular pathogen action. So it is used to design personalized medicine, minimize the side effects, maximize the drug efficacy. Okay, another one is quantitative genetics. It deals with traits controlled by multiple genes, that is polygenic traits. So number of genes are influenced in the particular mutation or in a particular phenotypic character, how the traits are measurable. Example, height of the stem, weight of the particular organ, or how much milk yield. Use these statistical methods, and it is vital in plant and animal breeding. That is called quantitative genetics. So these are the subdiscipline branches of the genetics. But how the genetics evolved from classical to modern, because number of organisms are help in the research programs. So we are using some particular specific microorganisms and other mammals or any uh, organism which is widely used in the genetics for the research, because they are easy to maintain, reproduce quickly, and have well mapped genomes. So you know very well this type of what organisms you are studied in at different chapters. మీరు